The first step of glycolysis is remarkable as this is the first irreversible reaction that occurs in glycolysis, and it is catalyzed by two possible enzymes. So in this video, when you carefully watch in details, you will see how one of the enzymes is connected with insulin, one is induced by insulin and one is not induced by insulin. And this is how insulin can cause diabetes mellitus when it is deficient in our body. So this particular step is very necessary. That's the step 1 in glycolysis, as it marks the first control step, and the enzyme involved in this step is one of the 3 enzymes that controls the rate of glycolytic pathway. So happy watching. God bless you. Don't forget to subscribe. I love what I do. I'm happy doing this, but I cannot do it without your support. Subscribe and share for others to learn.
Okay, we will see now the glycolytic pathway, the 10-step reaction in glycolysis, step by step. We will take step 1 and discuss it in details, step 2 in details until the last step. It will help you, you cannot fail again. So I hope you have listened to the summary of glycolytic pathway and also the song that I gave you that will enable you understand it. So let us now see how these things are explained, how come about this we are saying. So first step, step 1. But before I move into this explanation, because this will involve structures also, you should know that glucose exists in pyranose form inside the cell, pyranose, in form of pyranose, whereas fructose exists in form of fructose. So we must understand what pyranose means and fructose, what fructose means. What is pyranose? A pyranose is a cyclic organic compound that is made up of 6-membered ring. 6-membered ring, a kind of this, and this, just like you have your benzene, 6-membered. So we have 1, 2, 3, 4, 5, and 6. Then not only that every 6-membered cyclic compound is a pyranose, no, 6-membered ring that have 1 oxygen, then 5 carbons is what we call pyranose. So this is the form in which glucose actually exists inside the cell. So glucose usually take this form. Then fructose is a cyclic organic compound that have 5 members. 5 members, they are heterocyclic. When we say hetero, they are not of the same carbon. They are not of the same element. You have oxygen, you have carbon. The same thing, fructose is also heterocyclic compound, but it's 5-membered, but this one is 6-membered. So what we have here, you have a structure like this, such that when you now divide, you find out that we have oxygen, carbon, 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 and carbon. So this is a fructose, which has 1 oxygen and 4 carbons. So any heterocyclic organic compound that is 5-membered, out of which 4 are carbon atoms and 1 is an oxygen atom, we call it fructose. In the cell, fructose exists in this way, glucose exists in this way. But we might be seeing their structures in an open chain, depending on how I want it. So some of their reactions are actually involved in open chain form. Okay. So this will enable you understand the structures of glucose. So while drawing glucose in a cyclic way, this is the way it should be. Then fructose should be in this form. Step 1 in the glycolysis actually involves, step 1 involves the absorption of glucose by the cells and the subsequent phosphorylation of the glucose to glucose 6-phosphate. That is the first reaction. So structurally, we have the structure of glucose in this way. So in that aspect, we have H, OH, no, it should be C. So we are coming to that. You have 1. This should be 1, 2, 3, 4, 5. Okay, this should be only H, OH, OH, H, then you have OH for alpha hydrogen, that's for alpha glucose, then H, then here is carbon number 6. So this is the glucose molecule, then plus a phosphate group, which is actually in this form to give you this structure again. It will be phosphorylated. Numbering this shows that this is carbon 1, 2, 3, 4, 5, and 6. So the phosphate group will now be added to this carbon number 1. And what we have now, we have this oxygen, so here we now have our O, P double bond, O and O is having a charge of 2, then our H2 remains there. So these ones, hydrogen, OH, hydrogen, OH, hydrogen, OH, then OH, hydrogen. Please, while drawing this structure, you can exclude this carbon and just use sharp edges, which represents that carbon is there. So this is still accepted. But please, this is alpha glucose, that's alpha, in the sense that the OH, this is carbon number 1 and 4. The OH is down and down. Don't put OH here and H here. This one should be beta glucose, and it doesn't take part in this phosphorylation. Now, the enzyme involved here is either hexokinase or glucokinase in the liver. Now, to explain things in this step, first, you say that absorption of glucose and the subsequent phosphorylation of the glucose to glucose 6-phosphate. You can now see that this phosphate group is now added to this particular carbon number 6, and that's why we call it now glucose 6-phosphate. That is the name of this now, glucose 6-phosphate. That's just a glucose that have a phosphate group at carbon number 6. This is 5, 4, 3, 2, and 1. A glucose that has a phosphate group at carbon number 6 means glucose 6-phosphate. Now, what must we learn here? There is a remarkable thing. When you come to the liver, glucose is absorbed freely. But when you come to other cells, like the cells in the muscle, skeletal muscles, cardiac muscle, in your diaphragm, at the adipose tissue, insulin facilitates the absorption of glucose. But in the liver, absorption of glucose is free. It's not facilitated by insulin. You should note that. Then, it is at this stage also that you have to know there are 2 enzymes that catalyze this reaction. These 2 enzymes is hexokinase and glucokinase. The name kinases are actually a group of enzymes that are responsible for a phosphorylation. Responsible for phosphorylation. That's addition. Kinases means group of enzymes that are responsible for phosphorylation. That is adding a phosphate group. Then, from where did this phosphate group come from? This phosphate group came from ATP. That is, this phosphate group actually was taken away from ATP, such that ATP now is converted to ADP. ATP just means adenosine triphosphate, meaning it has 3 phosphate groups. Then removing that phosphate in it to add it to glucose, you have actually converted the ATP to ADP. It has been 3, now you have reduced it, remains 2. So, and there is presence of magnesium ion. You should note that the source of the phosphate group at this first stage is adenosine triphosphate. So the first step consumes energy. Then that means there is a hydrolysis of ATP to ADP, and ADP means adenosine diphosphate at the first step. And I told you that kinases means groups of enzymes that are responsible for addition of phosphate group to a substrate. Our substrate here is glucose. It is this enzyme, hexokinase or glucokinase, that catalyzes the addition of this phosphate group to the carbon number 6 to give us this. So at least we are getting the details in step 1. Then another detail you need to know is the differences. We have 2 kinases, 2 types of kinases that are responsible for this stage. And remember, please, This is the first irreversible reaction of glycolysis, and it also marks the first control stage. This enzyme is one of the enzymes that controls the rate of glycolysis. Now, the two enzymes involved are hexokinase and glucokinase. You should know the differences between glucokinase and hexokinase. Hexokinase are groups of kinases that just catalyze the phosphorylation of any 6-carbon compound. Any 6-carbon compound. Remember, this is total carbon here is 6. C6H12O6. That should be this glucose. So, and remember, there are other 6-carbon compounds, 6-carbon sugars, apart from glucose. So, hexokinase catalyzes the phosphorylation of any 6-carbon sugar. But glucokinase is now more specific to glucose only. Glucokinase only catalyzes the phosphorylation of glucose, not any other hexose sugar. So glucokinase cannot catalyze the phosphorylation of fructose. It can only catalyze the phosphorylation of glucose. Then, coming to their differences between hexokinase and glucokinase. If you come to our body, remember they are similar because their job is just to add phosphate to a group, and they are only adding to 6-carbon compound. The only thing here, first difference is that we have hexokinase, then we have glucokinase. This is non-specific, which means it catalyzes the phosphorylation of all the hexose sugars. I'm breaking this down such that even a layman who is not a biochemistry student will understand that. So enjoy it. This one is this is specific only to glucose. Simple. Another difference is this. Number 2, hexokinase is found in almost all the body tissues, found in almost all the body tissues. While this one, glucokinase, is only found in the liver. Found only in the liver cells. Then, number 3, this is found both in adults and found both in the fetus and adult, found both in the fetus and adults, while this is found only in the adult. Found only in the adult liver. Only in the adult liver cells. Then these differences will help you understand your work. Number 4 is that hexokinase is allosterically, I'll explain, because we'll be using the allosteric inhibition activation, so I will explain it here. Allosterically inhibited. Allosterically inhibited by its products. Its product is actually glucose 6-phosphate. Inhibited by glucose 6-phosphate. Permit me to write it this way. Then glucokinase is not inhibited by glucose 6-phosphate. Now, when I say allosterically inhibited, how do glucose 6-phosphate inhibit the activity of hexokinase? If you come to an enzyme, let's assume this represents the structure of an enzyme, where here is the active site for the substrate to bind. Then here we call this place the smaller modified site, allosteric site. What binds in the active site is actually the main substrate. The main substrate binds at the active site of an enzyme, taking this as an enzyme. Then if you come to allosteric site, what binds to it are actually effectors. Effectors binds to that. So effectors are any molecule, small sized substance or molecule that binds to a protein. Remember, enzymes are protein in nature. That binds to a protein to affect its biological activity. That is, it will bind, whenever this particular effectors binds only to allosteric site, substrate binds only to active site. So if you come to an enzyme, there is an active site and there is also an allosteric site. For allosteric site, effectors binds. For active site, the main substrate binds. Then, what is an effector as far as biochemistry is concerned? An effector is any small molecule that binds to protein to change its activity. It will either increase its rate or decrease it. So when the effector is actually decreasing the catalytic activity of the enzyme, we call it allosteric inhibitor. But when the effector is increasing the catalytic activity of the enzyme, we call it an allosteric initiator or activator, allosteric activator. As far as this hexokinase is concerned, the effector is the same product, glucose-6-phosphate. So if you come to hexokinase, GL6P, whenever it binds here, it stops the activity of this enzyme called hexokinase. First, it is the substrate. What is the substrate? The glucose will bind here. And the same enzyme will convert it to glucose-6-phosphate. Then when this glucose-6-phosphate is now high, too much, it will come and bind at the allosteric site. And that's what we now call it allosteric inhibitor. The same product of this enzyme is inhibiting the activity of this enzyme. But this particular glucokinase don't have the allosteric site for glucose-6-phosphate to inhibit it. So what do we mean here? Glucokinase is not inhibited by glucose-6-phosphate, but glucokinase is inhibited by its product, which is glucose-6-phosphate. Remember, the product of these two enzymes are the same. And what are the products? Glucose-6-phosphate are the products of these two enzymes. Now, that's it. So when I say it's allosterically inhibited, you should know that it is a small molecule called effector that is stopping it. So the effector here is actually the glucose-6-phosphate. So one can say that hexokinase is inhibited by its product, but glucokinase is not inhibited by its product. Then, another difference between these two is that hexokinase have high affinity for glucose, while this one have low affinity for glucose. Low affinity for glucose, which means that this particular enzyme in the liver can only bind to glucose when the blood glucose level is higher than normal. So the main reason for this enzyme to bind to glucose is just a kind of trying to make sure that there is no excess sugar in the blood. The main activity aim of glucokinase is not to yield energy, rather to go and convert glucose to a storable form, probably glycogen. But just note this difference. This one have high affinity for glucose, this one have low affinity. Almost divided by 50 of the affinity of this is equal to this. So the affinity of glucose to hexokinase is far more than the affinity of glucokinase to glucose. Cleared? Yes. Then, one should note this. There is what we call Michaelis constant, K-M. I think the Michaelis constant, you find that the Michaelis constant of this shows that it has high affinity for glucose, and this one have low affinity for glucose. Then, this will now lead to their main function. The main function of hexokinase is just to break down glucose for energy production. The main aim is just to degrade glucose for energy production in the tissues. Energy production in the tissues. Then the main aim of this is just to lower the blood glucose level, to regulate the blood glucose level. It regulates the blood glucose level, actually by preventing the excessive accumulation of glucose in the blood. It doesn't break down glucose to yield energy, rather it converts glucose to a storable form, converts excess glucose to glycogen actually. So it comes into play when just after meal and you have taken excess glucose, the glucose in the liver will be converted to glucose-6-phosphate. The aim is not to yield energy, rather to keep it in a storable form. In other words, this particular enzyme ensures that glucose is conserved in our body, not wasted when you have gotten so much. Remember, liver is responsible for regulation of our blood glucose level. How does it do that? I've just explained how it does that by using this glucokinase to break down the excess glucose. That's why it has low affinity. It can only attract glucose when the concentration of glucose is very high. But this attracts glucose even when it's very low, so that you can get energy. Because of the low affinity of hexokinase to glucose, that helps the brain and the muscle in case of danger. It will now make sure that the brain is giving more glucose and the muscles are giving more glucose. But in a very low energy level, it doesn't work at all. It is this one that works. That's hexokinase, converting it in a form that will give us energy. So at least if you have questions, ask it there in the comment box. Then coming to inhibition, we have actually number 7, I think. There are 2 inhibitors of this. Glucocorticoids and growth hormones inhibit the 2. Glucocorticoids and growth hormones inhibits the 2. Then if you come to inducible, there are what we call inducible enzymes and non-inducible. This is not induced by insulin. Hexokinase is not, that should be number 7, not induced by insulin. But the production of glucokinase is induced by insulin. Remember, that's why we say that insulin helps to regulate your blood glucose level. So if you have deficiency of insulin, you are indirectly trying to stop the production of glucokinase, and that will lead to accumulation of glucose in the blood, persistent high blood glucose level in the blood, which is called diabetes mellitus. So the work of this enzyme is just to prevent diabetes for you. Then this one is actually to give you energy. So we have seen that I said that both are actually inhibited by glucocorticoids and also growth hormone. But the difference here is that this is not induced by insulin, that's hexokinase, while glucokinase is induced by insulin. So it's an inducible enzyme. So there may be other particular, there may be other features that distinguishes them, but I've just given you the basic ones that your lecturer may ask you questions on. And I believe by the grace of God, I have detailed this. So if you like what you are watching and what you are seeing me deliver, please, I need your sponsorship. Subscribe, share my videos. And remember, if you are a student of biochemistry, you can have a private live stream with me for other metabolic pathways, of course. Then, at this stage, another fact we should know is that this glucose phosphate, this particular product of step 1, is an intermediate for other glycolytic pathway. We can see this also when glucose is going to be converted to glycogen. That's what we call glycogenesis. Even during gluconeogenesis, that's production of glucose from non-carbohydrate source. This is an intermediate. But whenever we have the glucose now being phosphorylated, that should be number 2. I wouldn't go there for now. You should just know that this product is not only an intermediate in glycolytic pathways, it's also an intermediate in other pathways, such as gluconeogenesis, glycogenolysis, and glycogenesis. We have this as their intermediate. So it's a junction between other things. And remember, I told you this is the first control. This enzyme hexokinase controls glycolytic pathway. Then, one should also know that glucose must be phosphorylated to glucose phosphate. Why? Because of the fact that glucose, when it enters into the cell, by normal diffusion, will still go out. Because I'm now explaining why glucose is phosphorylated to glucose phosphate. It's just a way of trapping the enzyme. It's a way of trapping glucose inside the cell. Because glucose, if this is a cell and this is our blood, this is the bloodstream, and this is the cell which is suspended inside the blood. And let's take a look at glucose concentration. Glucose concentration. Glucose concentration is always high in the bloodstream, and glucose concentration in the cell is always low. And as we all know, that in diffusion, of course, from higher concentration to lower concentration, there will be a kind of the glucose moving in. But what matters is that when it moves in, it will still be actively exported out. So the summary is that glucose is not stable inside the cell when it's in glucose form. It still moves out of the cell, still by diffusion or facilitated diffusion. whichever. So to stabilize it is to convert it to glucose 6-phosphate, which is not permeable to cell membrane. So glucose is permeable to cell membrane, whereas glucose 6-phosphate is not permeable to cell membrane. So why is glucose converted to glucose 6-phosphate? To trap glucose inside the cell. So glucose is found stable inside the cell only in the form of glucose 6-phosphate. Glucose 6-phosphate is impermeable to cell membrane, while glucose is permeable. So that's the major reason for this stage. So we have done justice to step 1 of glycolysis. So let's take a look at step 2. That should be next video.